So today we are going to discuss certain terminologies related to plant biotechnology. I think in this video we will not be able to cover all of them. So a second part will also be coming up. Number one is regeneration and embryogenesis. So regeneration in tissue culture means developing new organs or plantlets from tissues, callus or bud. Think of growing a whole plant from a small leaf piece in a petri dish. So this is a petri dish and here you have a small leaf, and from here you are going to grow a whole plant. It happens either directly without callus formation or indirectly via callus. Next we have embryogenesis. Embryogenesis is the formation of an embryo or plantlet that follows a pattern similar to natural zygotic development. In tissue culture, it occurs from somatic cells called somatic embryogenesis. For example, carrot somatic cells developing embryos similar to zygotic ones, producing many identical plants. Okay. Then we have in vivo and in vitro. You might have heard this term a lot of times. This is a Latin term which means in living. So processes that occur naturally in plants under field condition, that is seed germination in soil, root growth in the soil, these are in vivo because they are happening naturally under field conditions. Then we have in vitro. In vitro is also a Latin word which means in glass. It refers to controlled lab condition in tubes or dishes under sterile laboratory conditions. So example could be micropropagation in nutrient media or that shoot formation in tissue culture tubes. Then we have amplification and hybridization. Amplification is making many DNA copies using polymerase chain reaction, PCR, or natural gene duplication. So we can create as many number of copies as we want. So suppose we want to detect a virus in plants. So what we'll do, we will amplify its DNA fragment using PCR. PCR is basically polymerase chain reaction. We will discuss this in details in subsequent videos. Okay, next we have parasexual hybridization. Hybridization without sexual reproduction achieved by protoplast fusion. So here is an example. There could be fusion of tomato and potato protoplast to form a hybrid which is called as pomato. Okay, cell structures in tissue culture. So first one is protoplast. Okay, when we remove the cell wall of a cell, it is called protoplast. If there is no nucleus, that means enucleated protoplast, it is known as cytoplast. Then we have heterokaryon, cell with genetically different nuclei. Homokaryon, similar nuclei. Synkaryon is nuclei fused hybrid cell. Then we have cybrid. Cybrid is basically cytoplasmic hybrid. So you can call it cytoplast. You already know, right, what is a cytoplast? The second one, that is enucleated protoplast. Plus protoplast, this is known as cybrid. This is important, questions can be asked from here. Then we have heteroplast, cell containing foreign organelles. Okay, next we will move towards genetic engineering. Genetic engineering manipulates an organism's DNA to change its traits. DNA combined from two different sources forms recombinant DNA, rDNA. Okay. Plants carrying foreign genes are called transgenic plants. Very easy. Okay. So example could be BT cotton. It contains a bacterial gene for pest resistance. Right? Okay. Let's move to explant. Explant is small piece of plant tissue used to initiate a tissue culture. Then growth. It is increase in size or volume due to cell division. Differentiation, cells become specialized for particular functions. Okay, we'll talk about two more, that is de-differentiation. This is also important. Then we have one more, which is redifferentiation. For example, callus formed from carrot cells can redifferentiate into roots or shoots. So, callus cells form organs again. So, we will just try to sum up. Basically, differentiation means that cells specialize to perform specific functions, gaining unique structure and physiological property. So, if I had to give you an example, from animal, a stem cell differentiates into a nerve cell or a muscle cell. Then we have de-differentiation. Differentiated functional cells revert to a less specialized state, regaining their ability to divide. So in plants, parenchyma cell, once differentiated, can become meristematic tissue to form cambium. Okay. So this can happen during tissue regeneration, wound healing, or in the development of tumors where cells lose their specialized features and start dividing uncontrollably. Last one is redifferentiation. De-differentiated cells lose their ability to divide and become specialized again, maturing into cells with specific functions. For example, after de-differentiation, the cambial cells can divide and then redifferentiate to form secondary xylem and phloem in a plant. It is a subsequent step to de-differentiation where the regained ability to divide is lost as the cells differentiate into new specialized types. I hope now you have understood. Let's move to certain other terms. First one is callus. So, callus is an unorganized mass of cells from explant. Okay, I've already explained you both these terms. De-differentiation is mature cells revert to meristematic state, and redifferentiation is formation of organs from callus, basically again going back to it. Okay. Then we have one more term, totipotency. Every plant cell has the ability to grow into a complete plant called cellular totipotency. Example, a single tobacco cell regenerates into a full plant in culture. Then we have vitrification. Some in vitro grown shoots appear glassy, brittle and water soaked due to hyperhydration. It results in abnormal plants due to excessive humidity or gel strength. Okay, this is a term which can be asked. Next we are moving towards recombinant DNA technology, RDT. So the first one is linkers. Short double-stranded DNA sequences with restriction enzyme sites used to create cohesive ends for ligation. I hope you will remember. Next we have adapters, which is short DNA segments used to join DNA fragments with incompatible ends, used in cloning foreign genes into plasmids. So we also need to understand what are nucleases. So nucleases are enzymes that break nucleic acid. They break the phosphodiester bond between nucleotides. So they are of two types. First is endonuclease, which cuts DNA internally. Then we have exonuclease, which removes nucleotides from ends. Okay, they are also known as restriction endonucleases. So restriction endonucleases are enzymes that cut DNA at specific base sites. Right? They're differentiated on the basis of which side they're going to cut. So the term restriction endonuclease was coined by Herbert Boyer and Messelson. This is Herbert Boyer. Alright. In 1964, these enzymes are the molecular scissors of DNA technology, allowing scientists to cut and insert genes precisely. Then we have DNA polymerase. DNA polymerase 1, it is present in E. coli, is a DNA dependent DNA polymerase. It synthesizes a complementary strand using single stranded DNA as a template. Okay, so it requires a primer with a 3' hydroxyl group to start synthesis. So basically there will be a template and it will start copying it. Okay, this will help to make a copy, complementary strand. Suppose here T is there, so we will get A here, G is there, we will get C here, like that. Okay, it is used in DNA repair and in the nick translation technique to label DNA. Then we have terminal transferase. Terminal transferase adds nucleotide to the 3' ends of DNA molecule even without a template. It's purified from calf thymus. This enzyme is used to add sticky ends to DNA fragments cut by blunt end restriction enzymes. Example, adding a poly A tail to blunt end DNA before ligation. Alright, we have some more enzymes, that is alkaline phosphatase. This enzyme removes the 5' phosphate group from DNA ends. By doing this, it prevents the recircularization of the plasmids during cloning because ligase cannot join end without 5' phosphates. Example, used to prepare plasmid vectors for gene insertion. Then we have kinase. T4 polynucleotide kinase transfers the gamma phosphate from ATP to the 5' end of DNA or RNA. This process labels or activates DNA ends for ligation. So this could be used in end labeling, DNA fragments in genetic studies. We'll discuss all of these terms and details. So let me know in the comment section if you want a detailed video to come up on it where I will be explaining RDT to you. And we'll also come up with a part 2 because all the terminologies have not been completed in this part. I hope you have understood all the terminologies. Now here try to answer some practice questions. Number 1. Which of the following enzyme is responsible for adding nucleotides to the 3' terminal ends of DNA molecules in vitro? Number 2. The term pomato, a plant produced through protoplast fusion between potato and tomato, represents. Number 3. A double-stranded synthetic DNA fragment used to introduce restriction sites into blunt-ended DNA is called. Number 4. The process where mature plant cells revert to a meristematic state before forming callus tissue is known as. Write your answers in the comment section and let me know in the comment section if you want a detailed video on recombinant DNA technology to come up.